Anyway, Liam is a professor at Columbia of Statistics and Neuroscience, and he's the co-director of the Grossman Center for the Statistics of Mind. And he's done a lot of very important work, both in extracting signals from neural data. Some of you may use his algorithm CNMFE for extracting signals from one-photon imaging, calcium imaging data. And so we're lucky to have him here to talk about that and also analysis of data, and also like to welcome his postdoc, Matt. So thanks very much for coming, Matt Whiteway. Sorry, thanks a lot for coming, and thank you, Liam, for taking the time to talk to the class.
Thanks for having me. Yeah, so I'm gonna talk about our recent efforts as part of this International Brain Lab, and go through some of the fun parts and the not so fun parts we've encountered on the way. All the friends we've made along the way. Let me turn this back on so I can see you. Okay. Can you guys see this? Okay, everything cool?
Yes. Yep.
Yeah, awesome. Okay, so we started this collaboration about 5 years ago now, and when we got started, we're all very happy to be in neuroscience, and it's an amazing time to be working on the brain. But there are some obvious issues that were clear to us, right? So, for example, the large majority of our knowledge of the brain at single cell resolution has been obtained by these kind of fishing expeditions, right? Where you record from one brain area. Or maybe if you're doing imaging, maybe you can record from a couple brain areas. And every lab has their own little behavioral task that they like. And often maybe you're interested in just kind of one or a kind of a small handful of cell types, right? So it's a very kind of microscopic view of the brain. But of course, we know that behavior is kind of a brain-wide phenomenon. And so, to understand the brain, I think it's important to kind of zoom out and try to understand what's going on throughout the brain, ideally with single cell resolution. Of course, that's still hard, right? We don't have the technology to record from every single cell in the mouse brain, for example, simultaneously. So what we're going to need to do is pool data across many experiments, right? And kind of, by taking multiple snapshots of the brain, we're going to have to kind of figure out some way to obtain a coherent view of what's happening in the brain. But if different labs are doing different things and we don't have reproducibility across those different labs, that's going to make it not possible to pool these data sets and obtain a coherent view of neural computation. Another huge problem, of course, is that even if multiple labs were doing the same thing and the results were completely reproducible across labs, it's very much still not standard practice for labs to share all their data. Or if they do share their data, maybe they share just parts of it, and it might be hard to kind of understand quality control. Maybe some data sets are better than others, and you want to trust some data sets better than others. I sit in the Center for Theoretical Neuroscience here at Columbia. I'm very interested in applying all the latest and greatest kind of theoretical ideas to the brain. But it's very hard in general to combine theory and experiment at a deep level. So this was kind of the where things stood about 5 years ago, and we decided to kind of pull together a large collaboration to try to make progress on all of these issues. So we got together a really great group of both theory groups and experimental groups, about 20 labs total in the US and Europe, and we decided to kind of team up. So what are the goals of this collaboration, right? So the main scientific goal is to try to, again, understand what's happening during decision making at a brain-wide scale. Along with that kind of main scientific goal, we had some more kind of highfalutin sociological goals, right? So we wanted to really pull together experiments and theory is in as tightly linked a way as possible. We wanted to share all of our data and share all the both experimental and computational infrastructure that kind of underlie the creation of these data sets and these theories that we're developing, warts and all. And again, more pie in the sky. We wanted to, kind of change the way neuroscience is done by at least giving kind of an existence proof that large groups of scientists who sometimes like to compete with each other can kind of pool their efforts and work collaboratively with a big hug. So it's going pretty well. And I guess we're going to have a little philosophy section later, and I'd be happy to kind of talk more about the pros and cons of this approach. But that's kind of what we're aiming for here. Okay. So yeah, let's jump into kind of where things stand and what we've done. So Matt is going to walk you through some details about how to kind of take a look at some of the data sets that we've developed and some of the details of how we've kind of processed this data. But to get started, let's talk about the kind of shared behavioral task that we're using. So we decided very purposely to start very simple. We're using a very simple visually guided two alternative force choice task, where the mouse just kind of views a stimulus and churns a Lego wheel left and right to kind of match what's being shown on the screen with well, I'll just show you kind of what it looks like. So the stimulus kind of pops up here, and the goal of the mouse is to try to turn the wheel to the left so that the stimulus jumps into the center of the screen, right? So this would be a correct trial. Gets a bit of water, a little drop of juice for all these correct trials. This is a head fixed behavior, by the way. Here's an incorrect trial. The stimulus popped up on the right, and the animal kind of turned the wheel to the right, forcing the stimulus to go off the screen. So that's an unrewarded trial. Okay, so very, very simple task. We made it a little more complicated by modulating the contrast of the stimulus. So we had very easy trials, kind of harder trials, and we also threw in some impossible trials where basically the contrast was zero. So the animal had to kind of just make a guess about where the stimulus was. And I'll talk a little bit more about how the animal could use kind of prior knowledge to actually make that guess. So okay, so we got this task hardware set up in all of our experimental labs. And our first kind of data paper was about collecting data across multiple labs, lots of choices, lots of mice, and then trying to analyze how reproducible the behavior was across these different labs. Okay. And so if you look at psychometric curves, right, here are the psychometric curves from all the mice from one lab. You see there's again, this is the percentage of right choices as a function of contrast. So over here, the kind of easy trials to the right, the easy trials to the left, zero in the middle is again this kind of blank trial where the animal has to just make a guess about where the stimulus is. And so you see there's some diversity across the different animals within a lab, as you'd expect. If you look across multiple labs, you see, similar levels of diversity in these psychometric curves across the different mice. But if you kind of stare at these traces, it's hard to see that the inter-lab differences are greater than the intra-lab differences between mice. Right? There's kind of a similar amount of variability across labs as there is within labs. I should note that all of this behavior is kind of at the end of learning. So these are all with expert mice. Okay. And so we can play some games like, what if we try to build a decoder that takes the behavior of a given mouse and tries to predict which of the 7 labs this mouse is from. So you can kind of train that decoder, compare it to a shuffle, and you find that we were not able to build a decoder that got kind of good accuracy of figuring out which mouse the lab was from. Okay, and this data is available in this paper that came out a couple years now in eLife. Any questions on the task or the kind of basic approach to kind of testing for reproducibility here? All good? Yeah, go ahead. Or was that a thumbs up or a hands up? I couldn't quite see. All right, great. Awesome. All right. So that's you know, some very basic kind of quantification of the behavior. One of the nice things about this collaboration is we've been very interested in trying to push forward on kind of extracting as much information from the behavior as possible. So this is kind of another thing that came out of this collaboration. We noticed if you take a look at the choices that the mouse is making across multiple trials here, if you just keep track of whether the mouse was going to the left versus the right, whether the trials were correct or incorrect, just quantitatively, what you see is that it seems like the mouse goes through maybe a couple different phases, right? Sometimes the mouse is doing a great job getting mostly correct, following which side of the screen the stimulus is on. It's kind of engaged. Then there are some other kind of trials where we're trying to understand what's going on inside the mouse's brain as it performs its task, right? And if the mouse is a robot, it's going to kind of turn the wheel and kind of be completely engaged in the task until kind of the end of the day, and then it's going to go home, right? But of course, if you've ever played a boring video game, you'll know that your engagement level is going to vary as a function of time, and that's what we see with these mice as well. So if you take a look at the mouse's behavior as a function of trials here, remember this is just a binary behavior. So you can go to the left, you can go to the right, you can be correct, you can be incorrect. At this level of resolution, we're essentially just measuring 2 bits of information per trial here. And so during this kind of episode right here, during these 40-ish trials, it's clear that the mouse is really kind of biased to the right? It seems to make way more rightward choices than it should be. So you might say, well, maybe the mouse is not really paying much attention here. It's just kind of always spinning the wheel to the right. Maybe it's checked out. Maybe it's misunderstanding what's going on here. Who knows? And then later in the session, the mouse kind of locks in a little more, starts getting more correct trials, and has a nice balance between left versus right behavior. So if you just look at this, you might conclude, well, maybe the mouse is going through a couple different kind of discrete states. And maybe if we write down a simple kind of hidden Markov model for those states, we can actually extract those states directly from the data, right? And that turns out to be something you can do. The details are in this paper. And actually, Matt is going to walk you through some examples of this later today, where you can kind of grab some of this data and start to fit some of these hidden Markov model, HMM kind of models to the data directly. Okay, so we'll come back to that later in the afternoon. Did you catch that time? All good? Any questions on this one? All right, super. All right, so that's kind of a couple of the behavioral projects we've been focusing on. And there's more we can discuss afterwards if people are interested. But let's switch gears and talk about some ePhys data, some electrophysiological data. So as you all know, there's lots of different ways of recording from brains. We've been using kind of a combination of both calcium imaging and neuropixel recordings. I'm going to focus today on the neuropixel recordings. So the first thing we want to understand is, again, just as we did with that first behavior paper, is how reproducible are electrophysiological experiments across labs, right? So the way we tackled that question was to basically make repeated recordings, repeated neuropixel penetrations across multiple labs that targeted the same brain region each time. And then we analyzed the data and tried to study the reproducibility of those spike train recordings that we got from these penetrations. Okay. So just to kind of get you oriented, these repeated site penetrations are going to target these brain regions right here. So here's the cortex, hippocampus, thalamus, midbrain. We're going to be kind of targeting these brain regions. We collected on the order of about 100 or so of these repeated sessions, and I'm showing here the histologically reconstructed locations of these probes over those multiple sessions. So again, this is over multiple mice, multiple labs. You see there's a fair amount of heterogeneity in these probe locations. They're not all hitting the same kind of part of the brain every time. There are differences in kind of the angle we go in. Sometimes we have to move the probe one way or another to avoid a blood vessel. Sometimes we miss altogether. So there's as always, there's going to be some heterogeneity in these data sets that you should know about. But overall, we collected enough of these kind of penetrations going kind of near the same brain region that we have a pretty solid data set to study reproducibility here. So what's the kind of high level conclusion? If you stack all of these penetrations together and look at features like LFP power or firing rates or spike shapes, spike heights, we basically tried to think of a lot of different kind of electrophysiological features, plot them all and kind of do analysis with them. Basically, the conclusion is if you're sufficiently kind of careful with your quality control, you can achieve reproducibility across labs. So this plot is kind of a summary of one of those analyses. Each of these blocks here corresponds to one neuropixel probe, and we're just plotting basically the power spectral density at each location along this probe, right? And remember, each of these probes has been kind of localized by histology, and we basically tried to align all these probes next to each other to see how things look across these different brains and labs, right? So our friends in Berkeley collected 5 of these experiments. Our friends in Portugal collected about 9 of these experiments. We can stack all these together and then play similar games as we did before, right? If I give you kind of a random probe here, can you tell me which lab it's from? And if you can't, then that's an indication that our recordings are kind of reproducible across these labs, all right? So you could play exactly that game, train a decoder, see how well it does. And what we found is that we couldn't basically we couldn't build a decoder that does a good job at solving this kind of classification task. The decoder couldn't tell which lab we're recording from. However, if you use all the recordings without applying any quality control, what we found is that there were enough outliers, enough kind of weirdos in these data sets that you could start to actually decode the identity of the labs, right? So the conclusion here is, unsurprisingly, it's important to be careful with your data and throw away bad data sets, or at least triage away bad data sets. We developed some guidelines for this that are in the paper. This paper is available on bioRxiv as well. Any questions on these analyses? Yeah, go ahead. Yeah, that's a great question. So a lot of this is really just putting into place common sense kind of things that I think most electrophysiologists would agree kind of makes sense. So for example, we had 1 or 2 sessions where there was clear epileptiform activity that we were recording from on the probe, right? So that one got put in the box. There were some sessions where the noise level was too large, or where we didn't record really enough spikes across the probe, right? So we put in kind of a threshold for how many, what the minimal firing rate should be across the probe. Some of the recordings were very unstable. The probe was visibly moving over time in a way that we didn't feel we could kind of correct very well. We also put decently stringent kind of metrics on each unit, right, in terms of its signal to noise, in terms of its refractory period violations. So if you put enough of those together, you're going to start to kind of drop some sessions that don't don't achieve these controls. And so that's kind of the story here. Does that make sense? Cool. Any other questions? Yeah, so each lab kind of performed the experiments on their own. Of course, we're all using the same version of the NeuroPixels probes. We're using the same hardware across labs. This is open source hardware that anyone can build. The same kind of task software and so on. But the labs kind of collected the data independently. And then we pooled all that data, and then we had kind of a task force within the collaboration analyzing the data, processing the data, and trying to address these kind of quality control and reproducible kind of questions. So that's how we kind of organize this work. Did that answer the question? I suspect not. We didn't actually run that experiment. Other people have done similar things in the bioinformatics literature or in the fMRI literature, and found that there's a huge number of degrees of freedom in analysis. That different labs will do things kind of wildly differently, and that definitely hurts reproducibility. In this case, we had the luxury of being kind of in close communication with our partners in the collaboration. So we tried to get rid of that kind of degree of freedom. But yeah, that's a big issue. Other questions?
This may be a technical question. We find in our lab it quite difficult from the histology to determine the exact depth of a neuropixel penetration. Do you have a solution to that in terms of histology, or do you use the recording itself to reconstruct relative depth?
Yeah, that's critical. So we have a paper on this I can point you to. I wasn't planning to go over those details today, but in brief, we have a centralized histology facility at University College London. So every lab performs its experiments, then takes the brains out, ships them to UCL, where the imaging is done at this kind of centralized facility. And then after, I'm going to show a couple pictures later about how we look at the data and visualize the data. So we'll come back to this. But in brief, we have kind of a nice GUI that the people in the group have developed. There's kind of an automated portion where we kind of try to localize the probe track onto the Allen CCF. That's kind of a first automated step. And then there's kind of a second kind of quality control step where two researchers independently look at features like firing rates or LFP power, so on, and try to kind of refine the locations of those probes a little bit. And then we kind of look at the results of those two analyses together. And if there's kind of an agreement between the two aligners, then we say, okay, that looks okay. If not an agreement, then there's kind of further discussion. So there's certainly places where I think the alignment could be improved, but that's kind of the best we've been able to do so far. But yeah, that is a challenge. One of the kind of large scale projects we're working on now, which I'm not going to go into much detail, is trying to develop more automated methods that can basically take electrophysiological features along the probe and try to map that automatically into brain regions, using the fact that we have many recordings at this point from throughout the brain as kind of a training data set. So hopefully I'll be able to share some more news on that one maybe next year once we have some more solid results there. All good? Any more questions? Okay, super. So let me go ahead. So again, the take-home message here is you definitely need to kind of chop out some data sets. But if you get rid of some kind of obviously bad data sets, you're left with pretty good reproducibility across labs here. Okay. One of the big questions here is we see maybe going back to this picture of the behavior, we see quite a lot of within lab variability here. We see that on the behavior side. We also see it on the ePhys side. Certainly, even within a lab here. Thank you. You too. Certainly within a lab, even take these UCL labs recordings. There's some variability within labs. So one of the big questions here is, can we decompose that variability into different sources, right? Maybe some of this variability is due to just different behavior in these different mice. Maybe some of the variability is due to slight differences in where the probe went in. Maybe some of the variability is due to, different experimenters doing slightly different things, right? So it'd be nice to, put all the possible different sources of variability into a box and run some analysis that tries to assign variability, tries to decompose the variability into different kind of factors, right? So how would we do that? Well, one kind of standard approach, I guess you guys already know all about kind of encoding models, is to fit kind of a linear encoding model or a GLM or something like that to the data, right? So take all of your features, throw them into a GLM, and try to predict the firing rates of each of the neurons in your database, and then basically look at the weights of the GLM and figure out how much variance each of those features explain, right? That's kind of one standard approach. Of course, you need to know which are the right features. Also, linear encoding models kind of make very strong assumptions about how features will affect different sources of variability. So I think that approach is great, but it's got some pretty clear limitations. A different approach would be, we live in a world of artificial intelligence and neural nets. So why don't we use modern tools, right? Try to train a neural net to model the variability of each of these neurons. That's great, but neural nets require a lot of data and a lot of compute, right? So we probably don't want to just fit a different neural net to each of our neurons, right? That's probably going to be overkill, and we probably won't have enough data to kind of do that in a stable way. So one of the questions we came up against as we were performing this kind of reproducibility analysis is, can we learn a good set of features for the whole neural population in an automated way, right? So how balance the good parts of both the classical kind of GLM analysis and also the newer, fancier neural net analysis, right? So here's the approach we decided on, and we're not the first to kind of take this kind of approach. It's been applied in a few other brain areas, but it turned out to be kind of the right tool for this job. So here's the idea. We're going to take all of the covariates we can think of, which lab the unit was recorded from, what the behavior was on that trial, where in the brain the unit was recorded, and so on. Basically anything we could think of. And some of these covariates were kind of static across trials. Some of them were kind of more dynamic across trials, right? So for example, we're using deep lab cut to record the pupil diameter and the paw location as a function of the trial. So those are highly dynamic features within the trial. All right, so take all of these features we can think of, throw them into a neural net, and train the neural network to essentially extract a shared set of features across all of the neurons, right? So there's some kind of magic going on here, and the output of this neural net magic is basically a shared set of features. And then we basically fit a GLM for each of these features, all right? So that's kind of what the architecture looks like. Learn a shared set of features across the full population, and then individually, once you have those sets of features, train a standard GLM to predict the firing rate of each neuron. Okay, so of course you can kind of do both of these steps simultaneously, right? Update our kind of feature weights, then update the GLM weights, and run this until you have convergence. And so this kind of approach was very useful as we were kind of trying to figure out which covariates, which features predicted the most variance in our population. And again, the conclusion here was that if you compare this model with the lab ID included versus was not included, we found essentially no difference between the predictive accuracies between those two models, right? So the conclusion there again is that lab ID does not carry much information about the neural firing rates that you couldn't have gotten from other things like where in the brain you were or what the behavior was in each trial. Does that make sense? Okay, so again, that's kind of a nice tool that I think these kinds of questions come up all the time in neuroscience, and this kind of multi-neuronal kind of framework is useful for analyses that we'd like to do. All right, let me yeah, go ahead.
Give us an example of what can you give us an example of what's on each of the two boxes at the far left when you say static covariates or dynamic covariates? Can you give us a couple of examples?
Yeah, sure. Okay, so a static covariate could be which lab was the data recorded from, right? So just do a kind of a one-hot vector of which of those 9 labs you were taken from, or it could be where in the brain you were, right? Both of those covariates would be static across the whole session. Then there are things that are kind of constant within a trial, like the choices the animal made or the stimulus location. Those are kind of constant within a single trial. And then there are covariates that vary within a trial, right? Like the wheel speed is varying across the trial, or the pupil diameter, or the paw location. Those are all kind of dynamic within trials, right? So we had those kind of 3 different kinds of covariates: things that were constant across sessions, things that were constant across trials, and things that are changing even within the trial. Is that clear?
Yes, that makes sense. And just to clarify, both boxes could have continuous and binomial variables, right? Or one of them.
Yeah, exactly. So some of these covariates are discrete and some of them are continuous.
Okay, great. Thanks.
And we didn't do anything fancy. We just threw in all the covariates and let the model sort it out. Again, in the paper, there's some controls we did to kind of go ahead and check to make sure that if there was a lab specific encoding of these variables, could we actually detect it, right? And so we found that if you did put in kind of differences in the neural firing rates that were kind of lab specific, we could detect those with this analysis. Anyone else? All right, yeah, let me go ahead. Okay, so now we get to again, the main point of all this was to try to collect data from multiple labs, check to make sure that the labs are kind of collecting reproducible things, and then pull everything together to kind of try to map out what's happening in the mouse brain during this very simple decision task. Okay, so where are we with this kind of project? At this point, we've collected several hundred kind of probe insertions across many mice and about a dozen laboratories, and all of this data has been released, and we hope that many of you in the audience will take a look at this data and use it to pursue your own scientific questions. So let me kind of walk through kind of where we are with this. Here's kind of a fun cartoon of what the data looks like. Again, if we perform these histological localizations and figure out where in the brain each of these units were recorded from, color them based on their brain region, and then basically make these dots bigger or smaller based on the firing rates within the trial, we get a video that looks like this. So you can see here that the large majority of the recordings were targeted to one side of the brain. But we have pretty good coverage at this point across most brain regions within this hemisphere. There are still a couple gaps in the map that we're still filling in, but at this point I think we have something like 90% coverage. Okay. Yeah, go ahead.
So if I understood the statistics on the
Is there a
previous...
question?
Yeah, can you hear me? If I understood the statistics on the
Yeah.
previous slide, only about 10% of the recordings were good quality data. That means you're throwing out 90% of your data, which might raise some eyebrows. And the question is, if instead of trying to extract only the data which are of good enough quality to be synthesized across labs, you let each lab use their own quality control metrics and come out with their data, and then run your neural network feature extraction and have them then produce the GLMs, such that each lab now is not throwing out 90% of their data. Maybe they're throwing out, 50% of their data or 30% of their data, so that the data gathering exercise per lab is much more efficient. Do you see that the performance of their GLMs and the features that they come out with and the covariance explained by each feature are that different from what you get by first throwing out 90% of the data and then doing all of this only on the 10% of the data that meet the exclusion criteria across all of the labs?
Yep, great question. And yeah, it's a critical question. So a couple, one thing to clarify. First, we're not taking 500 sessions and then only keeping 50 of those sessions, right? We're taking 500 sessions and then keeping a small fraction of the units obtained from kilosort on each of those sessions. And yeah, I think there's differences of opinion about where we should set the threshold of good quality. I tend to be a little more conservative. Some pictures later, but you can certainly find some neurons even in this kind of good set, which are clearly either not single neurons, or there's some weird kind of non-stationarity that you wouldn't want to see. Conversely, there's certainly some good units in the pile that we threw away. However, there are also a lot of bad units. There's it's well known that kilosort and all current spike sorters make lots of weird mistakes that are not always easy to understand, why they're doing what they're doing. So I'm actually pretty comfortable with where we set this threshold. But of course others can disagree. So regarding the analysis of comparing what we can do with kind of this heavily quality controlled data set versus the raw data, this is something we haven't done what you suggested, which is perform this kind of GLM based analysis with all the data versus a fraction of the data. What we have explored is decoding analyses, where we say, okay, can I decode the choice or the pupil diameter or the paw location given all the data versus just a small fraction of the quote good data. And unsurprisingly, there's a whole literature on this in the brain-computer interface literature. Unsurprisingly, you do better if you kind of use the full data. So I certainly think there's more information here that we're kind of leaving on the cutting room floor. And I think improving on spike sorting is one of the the critical bottlenecks that we as a field have to kind of grapple with. But that's kind of where we are now. I would be thrilled. One of the big that we have as a collaboration is to see papers come out that we had nothing to do with, right? So we released this data. We hope that others kind of start to perform these kinds of analyses that you just suggested. It's not something we've done yet. I think it's all we'll do in the near future. So yeah, that's a great question. Other questions? All right, super. Yeah, so I think this video is cool. It shows lots of things going on in the brain, but there's lots more where that came from. So yeah, thanks for that question. So let's dig in a little bit on some of the kind of basic analyses that we've done, right? So we've focused on kind of quality control, and then kind of we've kind of purposely started, after collecting this data with trying to perform just a basic set of analyses that any neurophysiologist would want to see done. So just to kind of orient you, the way we're going to be kind of viewing these results is we've kind of unfolded the brain and kind of laid it out in this kind of flat map that's shown here, right? So of course the brain is a three-dimensional object. It can be hard to kind of view results in 3 dimensions. So we're just going to kind of unfold the brain and look at the results this way, and then we can do things like, all right, let's take all of the cells we recorded in the caudate, for example, and try to decode different kind of task parameters and see how informative those neurons are in this brain region about those task parameters. I've got a truck going by here. Sorry about that. Or let's fit a GLM to all the data in the superior colliculus, and try to understand which behavioral covariates are most informative about firing rates in the superior colliculus. So really just some basic analyses. And for example, if we apply that choice decoding kind of analysis to the neurons recorded from each of these brain regions, we can create a map that shows which brain regions seem to be more informative about the choices the animal is making compared to others. And what we see is that choice information, probably unsurprisingly, is kind of very highly distributed across the mouse brain. Lots of different regions have at least some information about the choice. And maybe part of this is because if the mouse is making different choices, that corresponds to different types of behavior. Or maybe if you make different choices, you get rewarded in a different way, right? So keep in mind that the task parameters here are not decoupled. There's strong correlations between the tasks, the parameters in this task. Also, just coming back to David's question, these numbers again are based on the quote good units only. If you allow yourself access to all of the electrophysiology on each probe, you can actually bump these numbers up significantly. So that's worth keeping in mind. So choice tuning is quite widespread across the brain.
Liam,
if you look at other variables, yeah, go ahead.
Liam, you mentioned about let's say training GLMs based on the data from each nucleus. Did I understand that correctly? Why not use all the data and then map using your spatial coordinates, do your clusterings like as a secondary step rather than train all the data from each individual nucleus? That's the first question. The second is like the differences in decodability between nuclei. Perhaps it doesn't look big. I'm not familiar how to read these differences of 0.1 or 2. Can you make a comment on these two, please?
Right. So I think for some of the details, it's best. This work is all in bioRxiv, and it's probably best to kind of point you to some of the details there on the preprint. We came up with 4 or 5 kind of basic analyses, right? One was this decoding thing. One was the GLM kind of approach. There was another kind of basic approach where we just kind of counted each neuron that seemed to be modulated by one of these variables or another. There are a couple others that I won't go into. So I guess in terms of the numbers here, the accuracies can be fairly low. Partially because in some brain regions, on a given session, we might not have recorded many cells, right? So there is quite a lot of kind of variability between regions in terms of the number of cells that we had access to. So maybe that addresses your question there.
Yes, it does. And one follow-up question, but perhaps we can push it to later, is the idea of flexibility of implementation, whether you have different ensembles and encoding things that if you would train over time, that would drop the performance of your decoder. Yeah, we can discuss this later, but this is just a thought I have.
Yeah, great questions. I think there's this is an incredibly rich and large data set. and I think there's lots of different things that I'd love to do with this data, that, you know, because, for example, we're busy trying to improve spike sorting, we haven't gotten to. So yeah, it'd be great to, you know, maybe chat in the next session about, you know, open ideas here and open ground. And again, I hope you all kind of take a look at the data and start to dig into some of these questions on your own.
Great, thank you.
Yeah, thank you. Yeah, let me go ahead and show you some more, right? So this is, you know, decoding choice. We can also try to decode the, you know, whether the trial was correct versus incorrect. Unsurprisingly, you see lots of brain areas that are interested in that variable. If you look at, you know, which side the stimulus came up on, again, lots of correlations there. One of the so I guess, you know, I guess the conclusion here is, you know, you can kind of make these brain maps using lots of different kind of behavioral covariates, and it's, again, just kind of a very basic starting point. You know, if you're interested in a certain kind of behavioral covariate, it's very useful, of course, to know kind of where in the brain you should be kind of, you know, focusing your attention. So this is now kind of available. Let me dig into something that's a little more subtle. So going back to the original slide about the behavior, I mentioned we had these trials which were kind of blank, where we had to kind of lean on the animal had to lean on its prior beliefs about which side of the screen the stimulus would be on. So let's circle back on that. So again, here's the you know, kind of schematic of what the behavioral task looks like. By the way, when should I stop? Because I think we started a little bit late.
Yeah, I think we can maybe go up till about 10:55 and then have a small 5-minute break before the next session.
Oh, all right, super. Okay, yeah. So this notion of priors, right? So here's the task, and we had some kind of easy trials and also some hard trials. So what is the animal going to do on those hard trials? Well, I didn't tell you the full details of the task. We've also actually broken up the task into several blocks. And basically these blocks are going to tell you how probable the stimulus is to be on the left versus the right. So the way the task is set up, we start with, it's not shown here, but we start with some kind of a 50-50 block where the, you know, the probability of left versus right is about 50-50. And then we switch to a kind of a left bias block where basically the and then we switch at kind of random times throughout the rest of the session. And the mouse, you know, is kind of able to figure this out. So for example, if we're in one of these left-facing blocks, the mouse is going to be biased on these blank trials to go to the left. All right. So this gives us a mechanism for probing how the animal is kind of weighing the likelihood, the data on the screen versus its prior information about, you know, which side is more likely. All right. And if we can kind of extract those prior signals from the behavior, then again, we can kind of map those back onto the brain using this brain-wide map kind of data set. Okay, so that's the game. So let's kind of walk through the details of this. And I guess we're going to be interested in, you know, two basic kind of scientific hypotheses here. One hypothesis is that, you know, basically, you know, prior knowledge is, you know, I guess if you go back to your Neuro 101 days, you think, you know, there's sensory visual information in this part of your brain, there's thinking stuff in this part of the brain, there's motor information about, you know, right around here, and these different kind of signals are distinct from each other, right? You might imagine that sensory information is kept segregated from prior beliefs in the brain until there's some kind of decision state where these kind of signals need to be combined, right? So that would be hypothesis 1. And hypothesis 2 is kind of the opposite of that, both priors and likelihoods are kind of widely distributed throughout the brain, maybe even in sensory areas or in other kind of brain areas that we don't really think of as sensory or decision related at all. All right, so these are the 2 hypotheses we want to address. Okay, so let's go back to the behavior and just check that these different blocks. All right. So if you segregate out the psychometric curves computed during the left blocks versus the right blocks, and average that across mice, what we find is that there are strong differences in the psychometric curves in these left versus right blocks, right? And in particular, there's a pretty big gap here in the 0 point, where again there's 0 sensory information about what the correct kind of decision would be, right? That seems good. We can also look at the dynamics. Plot the proportion correct as a function of time since the block switch, right? So let's look at this and try to understand it a little bit. So usually the mice are pretty good at performing the task. The proportion correct is pretty high here. There does seem to be a dip after the block switch when you go from 20% on the left to 80% on the left. But this effect kind of at first glance looks a little bit small. There's actually not a huge dip in the proportion correct. But then you remember that actually most of the trials here are actually pretty easy. They're going to be likelihood dominated. So what if we just restrict our attention to the prior dominated trials? And there, if you look at this red curve, you see a much bigger jump, right? So the proportion correct is much lower here because again you have 0 sensory information about what's the correct answer here. After a switch, your proportion of correct drops significantly, and then after about 10 or so trials, the mouse is able to figure, to or right. Now I'm kind of back in the game over here. And actually, you can do a little math in this task and define kind of the Bayes optimal model. Just out of curiosity to compare how far from optimal is the mouse's performance here, and they're in the same ballpark. And so the optimal model is able to figure things out a little more quickly, but the mouse gets there pretty soon. Is that all clear? All right. So that's behavior. Great. So let's switch back to the neurons. All right. So what we find when we perform this kind of mapping exercise again. So again, the plan here is to take that behavioral signal that we just extracted, use that to estimate, use that model to estimate basically what prior the mouse is operating on under each trial, and then run the correlations so that you can correlate this prior signal that we've extracted from the behavior back onto neural firing rates. And what we find is that about 30% of brain regions kind of show some significant correlations to this prior signal. In areas that we might have expected, and also in some areas that were surprising to us. So I don't remember much about the pawns from my neuroanatomy 101 a couple decades ago, but I would not have guessed that this brain region would be kind of strongly correlated with the prior. And of course there are multiple correlations here, both behaviorally and cognitively. But it's interesting to see how widespread this signal is throughout the brain. So again, this result, I think, is much more kind of consistent with the second hypothesis that we had before, that the prior information is not kind of segregated to a small brain region kind of far away from the sensory information in this task. Rather, it's kind of broadly available throughout the brain. Maybe not in the same cells, but certainly in the same kind of brain regions. Is there a question?
Yeah, I wanted to ask, when you determine whether a region significantly decodes the prior, does that already factor out the fact that, of course, there's going to be different activity because the proportion of trials is different? right? We already knew that you can see the difference between left and right trials. And simply from the fact that there are different left and right, different numbers of left versus right trials, you could therefore decode the trial state, which is not the same as there being an actual prior encoded.
Yeah. So again, I'm skipping all of the controls here and the details. I should have added a link to the paper. This paper is also up on bioRxiv now, so you can grab it. I can share that with everyone afterwards. But yeah, we did perform several kind of analytical controls to check exactly these kinds of issues and to try to decouple these behavioral correlations. But again, in the interest of time, let me just point to the paper for those details. Any other questions? Yeah.
When you say prior, can you please define it for us? Is it like a 5-second history? What exactly is it?
Yeah. Again, there's several slides here that I'm skipping, but yeah, one of the points of this paper was to try to use several different models to kind of estimate what the mouse's prior was, right? So this Bayesian optimal thing that I mentioned, that's one potential model that we could use. There are other kind of GLM based models where you look at the 5 recent trials and try to predict the next trial's behavior based on that, on the recent history. There are several other kind of Markovian kind of models that you can imagine. So we basically did a little bake-off with several of these model classes and tried to understand how much of the mouse's kind of behavior could be explained by these different models. And again, several of those models are discussed in this paper. And so we basically took kind of the best one and used that as the estimator of this prior that was consistent with the mouse's behavior. So that's what we're kind of correlating back onto the neural activity. You can use other things, like you can use the output of this Bayesian optimal and use that to perform the correlations, and you get kind of similar results. Again, because the Bayesian prior or the Bayesian optimal looks not too dissimilar from the real data in this case. Does that answer the question?
I see. Yes. Thank you.
Okay. So let me I think I'm low on time. So as always, I have lots more slides that I want to show you that I won't be able to. But let me just close by showing some of the visualizations that we've developed for this data. One of the things this project has taught me is critical it is to develop tools where you can kind of very quickly kind of address and look at the data in multiple different ways. And we actually have a visualization working group led by these three and several others who have really put together some amazing tools that I think could be generalized to other data sets as well. So let me kind of show you just for the last 5 minutes what these things look like. So you can grab these tools. This is available. Again, all of this data is downloadable from the very raw data to kind of the higher kind of process level. But let me just kind of click on this link now and show you what this visualization website kind of looks like, so you can kind of look on your own. So you kind of start here. And you can kind of select whatever penetration you want throughout the brain. If you're interested in those repeated sites, you can grab the subset over here. Or if someone has shared a data set with you and said you really need to look at this data set, you can just kind of type it in here or scroll through that way. So the first step is to kind of grab a data set and look at the metadata over here. And then you can kind of take a look at kind of an overview of what's available in this data set. So this block here shows the raster map. Essentially, each dot here shows the firing rate of one neuron located on the probe on the y-axis here as a function of time down here. You see the choices and the contrast that are being shown to the animal over these trials. We actually have kind of a passive part of the task. out here where the animal is not actually performing the task, just to get a little more data. Over here, you can kind of see where in the brain you were and what the kind of histology looks like for this recording. So this kind of gives you an overview, and already, you know, if you look at this overview, you can kind of see some stuff. You know, certainly the recording looks pretty stable for most of these trials, but something weird happens here. Maybe some neurons were lost. Maybe there's some kind of non-stationarity of the probe here that wasn't fully corrected. So again, this is very much a warts and all data set. There are things you'll see as you kind of browse around here that you know, we still need to improve further on. So that's kind of the neural data overview. We also have kind of a behavior overview where you can look at the psychometric curves for this individual session, look at things like reaction time as a function of trials. Here, you know, the animal kind of quits out here and the reaction times go way up. You can look at things like, you know, the pupil diameter, or the wheel velocity, or when the animal was licking. A lot of these are derived from Deep Lab Cut, and Matt is going to kind of walk you through some of the details of how we process that video data. You can kind of click around and find individual trials here, and look at the raster map on the individual trial level. Load this up. You can also look at what the behavior was on individual trials. So again, we're recording video from 3 different cameras here, monitoring the paws, the face, and the back of the animal. This video, which was randomly chosen, was one with pretty low contrast. Again, you'll see a lot of heterogeneity across these data sets, even though we kind of did our best to make each data set as reproducible as possible. And then finally, you can kind of scroll down here and kind of look at the individual neurons, right? So you can click on one of these units here. Sorry, let me try stopping that again, so I don't have to listen. So you can click on a random neuron here, and look at its firing rate as a function of time, or its firing rate as a function of kind of correct versus incorrect trials. Here again is a neuron. If you look at that neuron's amplitude as a function of time, you see that it's pretty stable for a while, and then something happens, and you know, we probably lost that neuron. So this, you know, is a neuron you should kind of be careful of, right? Click around and find other examples. Here's a neuron that was not present for most of the recording, and then it kind of popped up towards the end, right? So there's lots of things to watch out for as you analyze this data set. To be clear, I don't think these non-stationarity issues are kind of endemic to our data set. I think they're visible in any NeuroPixel data set that you can look at. But we've tried to make these issues as kind of visible as possible in the data. Any questions on this? Okay. So maybe that's a good place to pause. Definitely lots more to talk about, but I guess the plan is to take a break and then have a little ask me anything session afterwards.
That's good. Thanks, Liam. So 11:05. Okay, we'll take about 10 minutes.
All right, super. We'll see you all then. Thanks, all.
Thank you, Liam.